I want to talk about the kind of thing we do, including the data set that's on the Dandy archive, but also more generally kind of the way that the lab thinks about neuroscience, and I'll also included some new data sets. And I know that some people were interested in these data sets, so I'm trying to move my next meeting so that I have like an hour after this, so we can chat if anyone wants to. My PhD was my undergrad was in mathematics and physics, PhD in computational neuroscience, and at the end of my PhD, I went into experimental neuroscience because I felt that basically any theories or models were under constrained, and although there are emerging techniques from machine learning, for example, that would facilitate our theoretical understanding of the nervous system, the data wasn't there to constrain theories sufficiently. And that wasn't necessarily because there wasn't enough data. And it's become, at least for a while, I think not anymore, but for a while it was this mantra that we have too much data. We have so much, like, data is big and so on. That was mostly, like, it was just a lot of experiments, but I think not always the experiments of the right kind. And so I think the experiments of the right kind that we need, the critical thing is that they cover many modalities inside the brain and outside the brain. So here are some of the modalities. This is not exhaustive. So behavior, whole brain activity, connectivity, molecular information, dynamics across the body. Of course, many things are missing: voltage, hormones, the environment. But these are some of them. And so to make this more tractable, we're using the larval zebrafish because it's small and transparent. And even when it's very young, 1 week of age, it already performs a lot of interesting behaviors, including very fast forms of learning, motor adaptation, evidence accumulation, and so on, some of which I'll present here. So here's just some of our work in these different areas. And I think the critical piece is going to be about merging different modalities so that, because if you merge modalities, if they are informative about one another, what you get out is more than the sum of the parts. So that's been a lot of the kind of the technical goals of the lab. But as an illustration of, am I audible or too loud or too soft? You're good? Okay. As an illustration of kind of the way we're thinking and why we're bothering in the first place, I'm going to discuss two behaviors: positional homeostasis and futility-induced passivity. The second behavior is the one that's on Dandy. And both of these, but the critical piece is integration of information over time and execution of decisions over time, which is a hallmark of more complex biological systems. And then I'll talk about expanding the possibility of discovery, whole brain connectomics and function, and whole body imaging. And I hope I'll get to that. I have half an hour, right? So I'll try to go a little bit fast. So the first work's by N. Young. The idea behind this work is that many fish swim against the flow, and they probably do that to stay away from dangerous areas, including dry land. And they use largely optic flow to stabilize their position. So it's good to stay in the same location, because if you've been safe for the past hour, you're probably going to be safe for a while longer. So it's safe. There's another reason. If you let yourself be swept along with flow, then any kind of food particles that are around you are going to be swept with you at roughly the same speed, and you won't find much food. Whereas if you swim against the flow, you can kind of encounter new small animals to eat. So food. And why go? You may need to actively swim somewhere else, explore for more food, or hide from passing predators. Or there may be some fast currents that sweep you elsewhere. So we asked, can animals find their way back after such displacements, and how does the brain do it? So this is one example of like a slightly more complex behavior, because these are small animals with about 140,000 neurons, the same size of a brain as a fruit fly, or only slightly bigger. Like how complex is their behavior? So often we use virtual reality environments to probe the neural basis of behavior. So if you imagine the behavior you just saw, swimming against water flow, you can you can depict that as position over time in the presence of flow. In VR, we would invert that so the animal would be stabilized and the image of the ground would move. So when the animal swims, the ground gets accelerated backwards. So for multiple reasons, one is for imaging stabilization, and also for improving behavior, which sounds paradoxical, but it's true. We paralyzed the animal by blocking the neuromuscular junction, so the brain and spinal cord is still fully functional. But we use electrical recordings from the tail motor nerve to drive movement through VR environments. So in this case, we use fictive swim bouts to drive swimming through VR in 1 or 2 dimensions. So yeah, we call these fictive swim bouts. And here you see an example. The animal's over there. It's swimming around in a 2D environment in the presence of simulated flow. We always simulate flow using optic flow. So if the simulated water flow is backward, we simulate it by providing forward optic flow. We can now combine this system with whole brain light sheet imaging, where we use selective plane illumination microscopy that we developed, or we adapted soon after I started here together with Philip Keller, that allows us to get data sets like these at multiple brain volumes per second with about 90% of the brain at single cell resolution. We can do that using genetically encoded calcium sensors, which is the predominant modality that we look at, but also other sensors like neuromodulators, voltage, and so on. So coming back to the behavior that you saw before, one possible control system is that the animal perceives self-velocity through its visual system and swims, which changes self-velocity and could roughly stabilize its location in space. So this very kind of feed-forward system would stabilize the animal in space in the presence of water flow. So water flow triggers non-zero self-velocity. Swimming causes a change in self-velocity. And this could roughly stabilize the location of the animal. But if we do a thought experiment, we can see easily when this kind of strategy would fail. It would allow the animal to approximately stay during steady flow, but it wouldn't be able to return after unexpected displacements because it has no sense of memory. So if we have steady water flow, it would stabilize its location if the system is approximately tuned. But if there's, for example, a sudden brief current that makes the animal move forward before it can react to it, then the animal, if later on it starts swimming against a new steady flow, it would simply perform the same behavior elsewhere in space. So it has not corrected for any errors that happened in the past. So this kind of non-convergence of trajectories is a signature that the animal lacks kind of temporal integration abilities. So we call this the perturbation, the pause, and the correction, or the lack of correction. And so in this case, this kind of control system generates an error, which we can fix by mimicking certain aspects of a PID controller through a temporal integration that turns self-velocity into positional memory, which should allow the animal to return after displacements. And by position, I always mean position relative to the original position. And so in this case, a sudden displacement would trigger, this positional component of the control system, which would allow the animal to change its behavior in the future. For example, wait longer in response to future water flow and swim back to the original place. So in this case, there's a correction. If we contrast these two control systems, it turns out that behaviorally we can show that this one is pretty close to what the animal is actually doing. As you can see here, if we displace the animal forward or backward in VR by about 10 body lengths, and we introduce a pause period during which the fish is fixed into place and there's no flow, so the animal tends not to swim. And then during this correction period, the animal manages to get back to its original location quite accurately. So this second control system is close to what the animal does behaviorally, and it has a memory time constant of about a minute. So that's quite long for a neural integrator. So now going to whole brain activity measurements, this is a top projection of many planes of neural activity. if we have a good understanding of behavior through control theory, we can often, or we can try to identify the components of the control system in brain data using relatively simple measures. In this case, a form of correlation. Of course, this won't always work, and this is one of the challenges of analyzing these complex data sets, to figure out what to do if this doesn't work. And of course, even if it works, it doesn't mean that you have captured the whole story. So if we do this kind of correlation, we can find position encoding cells in multiple fish, here superimposed, in specific clusters in the brain. And the strongest representation of position is in this area, which we call self-location encoding medulla oblongata neurons, or SLOMO, for lack of a real name, although we think we know which area it is in mammals. But we haven't firmly established that yet, so for now we stick to our own name. And if we look at activity here, we can see neurons that encode forward displacements with an increase in firing rate, and backward displacements with a decrease in firing rate, and the reverse. You can see from these two example cells that, that this is not a pure representation of position of the animal. It's mixed in with other signals, like in cell 1, a ramping signal that starts ramping at the end of the previous trial because the animal stops swimming. When there's an offset in swimming, it starts ramping, but on top of that ramping signal, you see a positional signal riding on top. So this is positional integration because the positional signature is still present after many seconds after the positional shift. If we do dimensionality reduction through even simple measures like PCA, we can essentially read off where the animal is in space by looking across this diagonal. Okay. And so when the animal performs this spatial trajectory in PCA, it undergoes this network trajectory. And this also occurs in 2 dimensions and for complex displacements. So now how do we go from correlation to causation? We can combine whole brain imaging with large-scale online analysis and random access neuronal perturbations. I'll fly through this, but essentially we managed to test this model. And for example, here this is one example of what we did. We took out this positional integrator, and on the left you can see an intact fish that manages to make these trajectories converge on the different trial types. If we ablate this area, SLOMO, then the animal loses the memory component. It still responds to the stimulus, but you can see there's a complete loss of positional memory that's reflected by the fact that on different trials, these trajectories no longer converge. So we've turned a controller with a memory into a purely reactive feed-forward controller through whole brain imaging, analysis, control theory, whole brain imaging, and function-guided perturbation of the system. We can also then consider the other brain areas that we found contain positional signals to start working towards a multi-regional network understanding. And so now we have a fairly good understanding of the brain-wide circuitry underlying this behavior, but to reach a full understanding, we need to know the synaptic connectivity at the cellular level, which I hope I'll get to later in the talk. So this is our general strategy for interrogating brain function at the multi-regional level. So that was a circuit that allows the animal to stay in place during water flow. And now I'd like to turn to the DANDY dataset, which involves a switch in behavior when it's impossible to stay in place. So this is the behavior I talked about before, staying in place. So positional homeostasis, staying in place for multiple reasons, including food and safety. But this kind of behavior also requires a lot of energy. It's like the fish is running on a treadmill, which never stops. So sometimes it's better to give up and preserve energy. For example, when there are fast and chaotic currents that the animal couldn't possibly overcome. Or if the animal's stuck in debris, also a case where actions are useless if the animal's really stuck, and it may be better to wait. Perhaps the debris will wash off naturally and the fish is free again. Or under low temperatures, if neural commands from the brain that aren't sufficient to make the muscles contract effectively and make the animal swim against the flow. So Yumu, who's now a faculty in Shanghai at the Institute of Neuroscience, investigated this question when he was in my lab. So we call this kind of behavior swimming in closed loop because motor output has an effect on the environment, specifically the animal's position in the environment, and the animal's position feeds back onto motor output through the visual system. We can change this because in virtual reality, we're in control of what happens when the animal acts. We can change this to an open loop environment where swimming no longer has any consequences. And in this case, we simulate an environment where the animal can swim but doesn't move in response. So we open the loop, and in response to this, animals tend to give up, which is kind of a natural response that we would also probably do. So in closed loop, the animals swim pretty regularly for tens of minutes. In an open loop, they kind of flail. So in closed loop, they move forward. In open loop, swimming is useless. And in response, they tend to then suddenly give up. This is what we call futility-induced passivity, because it's behavioral passivity induced by behavioral futility. If you keep them in open loop, they don't give up forever, which would be kind of bad. So instead, they switch back and forth between trying and giving up. And fish aren't the only animals that do this kind of behavior. Here's a squirrel trying to get to food, but the pole's too slippery, so there's a sudden switch, which is unlikely to be triggered by an actual lack of energy. It's probably triggered by the brain in anticipation of a depletion of energy resources. Pretty what? Pretty close. Yeah, it's tragic. I really, but you forget that that the platform is also an additional challenge. So this was not me. I would not do this to the squirrel, but you can see that in both cases there's this vigorous struggle followed by a sudden switch to passivity. So this behavioral algorithm that I showed you before is clearly insufficient because this control system will just keep trying. So we need to expand it into essentially a failure detection where the consequences of swimming are considered. So swimming normally leads to change in position, but if this is not the case, then these failures should be registered by the brain. You also saw that these animals don't give up immediately, which is a good thing. You should keep trying for at least a while. And so there must be an integrator, which here I depict as a sum, but of course it's a temporal integration. And when this crosses a threshold or some other critical value, then swimming needs to be inhibited for some amount of time. And it's funny because there are two books written about like quitting. One is like quitting a way of life or something, and there's another one that's being written. And both of these books are about like why quitting is good. And so this is not part of the presentation. This is just like it happened in the past few weeks. One of them was published. They described this study plus a bunch of other studies, and another one's being written. But both of these authors were very interested in why quitting is good. And so, but I was kind of arguing with them because I feel like being scientists, it's still kind of we thrive on persevering. But I think that in society there's apparently an emphasis on always keep going and never giving up. So I guess the trick is to kind of find the right balance and to take your gut feeling of these integrated failed attempts seriously instead of ignoring them forever. So that was kind of the punchline that eventually satisfied the author. When her question was, what can humans learn from fish? So apparently that's what humans can learn from fish. So, but I would say that often you want this end to be not infinitely high, but high enough that you give yourself a chance in science. So again, with the hypothesized control system and behavior, we can now ask, given these two pieces, can we predict what we should find in the brain? And with these two pieces, we can ask, what would the failure signal look like? And this is what it would look like. It's 0 when the animal swims in closed loop or when it's not swimming. and it's non-zero if the animal swims in an open loop. And we can actually play with this. We can make the animal feel even worse and make the animal sense as if it's moving backward when it's trying to swim forward, even when it's worse than staying in place. So, or we can make the animal stuck, but make the animal try with different motor intensities, so that the harder they try, the bigger the failure signal is, and so on. So, but this is roughly what it looks like, what it would look like if this is correct. This integrator would look as like the integral of the failure signal, and the signal that inhibits this naturally occurring innate behavior would look like the blue curve over here, motor suppression. So we can, again, look for these signals. Beginning with failure detection, we can correlate whole brain activity to that, and we find a very specific set of neurons that are correlated with behavioral failures. And this is the result of a computational analysis of the data, which very strongly overlaps with this anatomical molecular volume. This is a transgenic label for cells that secrete norepinephrine, which is a neuromodulator known to be involved in things like alertness. And this labels the locus coeruleus, the LC, in an area called NEMO. And if we stimulate NEMO, we can trigger vigorous swimming followed by giving up artificially using optogenetics. And if we ablate NEMO, then we can produce animals that almost never give up. They just keep going. And if we do the same with the locus coeruleus, then if we ablate the locus coeruleus, then there's no change or very little change in giving up behavior. So we conclude that norepinephrine neurons in the hindbrain are the cell type that seem to be causally involved in this behavior. And just for clarity, this is different from the slow-mo cells that I described earlier. They're neighboring areas, but they have different neurotransmitters. Then for the failure integration signal, we had to go look beyond neurons. We had to also look at astrocyte activity because we couldn't find this ramping signal in neurons. And so again, we did something like a correlation, and this is the type of data that's on the Dandy archive. This is whole brain cellular imaging. Swimming is at the bottom. This is the current time, so the swim signal will be scrolling to the left. It's sped up by a factor of 3. Neurons are shown at the top and astrocytes are at the bottom. And here you see 5 slab projections, but this is actually 40 planes of imaging. And so they're collapsed into groups of 8. So in the archive, there are, I think, a few fish with double color imaging like these. And then there are other fish that have either one or the other. Because if you expose the animal to more light, the behavior isn't as good. So we also did a lot of experiments where we either image neurons or astrocytes. So here you can see that during vigorous swimming, neurons are on average more active. But during passivity, astrocytes are on average more active. But importantly, astrocytes already start ramping up during vigorous swimming if the animal is failing to move. So you can see this integration process right now in the astrocytes already taking place, followed by calcium maintenance during the passive period. So to summarize this movie, during closed loop, you see on the left that wasn't actually part of the movie. But this is a snapshot of closed loop swimming. You can see very little astrocyte calcium levels. This is open loop swimming. You can see the integration is starting, peaking, and then is maintained during the passive period. So after that, this shows that astrocytes integrate swim failures. You can, as I said before, you can play with the degree of failure to show that this is really true. If we now optogenetically or chemogenetically activate astrocytes, we can trigger passivity. Or if we silence astrocytes pharmacologically or through ablation, we can create fish that almost never give up. And so we conclude that astrocytes aren't just the integrators, they also trigger these behavioral state changes. Then we also found the cells that suppress motor output. These are GABAergic neurons in the lateral medulla. So this is the shape of this core circuit. Noradrenergic cells here, astrocytes, the cell bodies are here, but the processes are here in the lateral medulla. And they're GABAergic neurons with cell bodies in the lateral medulla, processes in the lateral medulla, but some of them also cross over to the other side. This is the core circuit. And so to summarize what I already described, the noradrenergic neurons detect behavioral failures. Astrocytes integrate this noradrenergic signal over time and trigger activity in GABAergic swim-suppressing neurons. So in this case, we could more or less match the control system to the circuit. But this won't always be true, of course. I think the better you understand the behavior, the more accurately or the more comprehensively you can build these control theoretic models of the behavior. Or maybe you need more advanced models, maybe control policy models or whatever. But the better the model for the behavior or the class of models you have for behavior, the higher the chances that you can interpret the neural activity. And maybe you can iterate between the two. So this is the website. And thanks again, especially Ben and Cody for working with us on that. So yeah, it's all available here. And then I guess I'll try to wrap up soon. A few more things. We also showed that ketamine has the kind of effect on this behavior that you'd expect from mammalian studies. If you expose the animals to a transient dose of ketamine, you can make the animals resilient to behavioral failures, and they tend to keep swimming and not give up. So this is what you'd expect from animal models, which are sometimes interpreted as models for depression, but really they're only a model for a very small, sort of tiny subset of depression, but they have been used for drug screens. And so what we found in this study is that ketamine, at least in fish, works by hyperactivating the noradrenergic astroglial system that I just described massively during ketamine exposure. Then you take the ketamine away, and for some reason, cell biological reason, presumably that we don't yet understand, this whole system has now desensitized. And that's why animals respond less to behavioral failures, and they become more perseverant. We also found the mechanism by which astrocytes communicate to neurons. So if you look at this circuit, we kind of figured out early on how the noradrenergic neurons activate astrocytes. That was more or less known in mammals as well, through which receptor that happens. But it was a mystery how astrocytes talk back to neurons. And so we found that happens through secretion of ATP and conversion of ATP into adenosine in the extracellular space, and reception through adenosine receptors on neurons. So there's more work we did here. Also other stuff I can tell you about, like the large-scale network effects of astrocytes. But just looking into the future, I just want to take like a couple of minutes to really kind of merge modalities and get to the bottom of how structure and function correspond, how these patterns of brain-wide activity emerge. In the first place, we wanted to look at brain-wide activity together with brain-wide connectivity in the same animal. And so this is work by Alex Chen, a very talented graduate student between my lab and Florian Enger's lab at Harvard, who looked at 9 different behaviors in the same animal. So this is a large collaboration. And we wanted to create a community resource of activity over time together with connectivity and behavior. So this is already available through ICLR. I think, we could also put this in NWV format, some preliminary clustering. But the important point is that we have whole brain activity with whole brain EM connectomics in the same animal matched cell by cell. At least we're working on this. We're matching, so we're making progress in registering and in reconstructing the connectome. So this is very much work in progress, but we have both this data for the same animal. So hopefully we can start to understand how the synaptic connectivity gives rise to these population codes. And merging these modalities, I think, will enable new opportunities for discovery through machine learning, computational neuroscience, biophysical network models. And again, the data is available online through ZapBench, which was led by Viren Jain's group at Google. We're doing the same thing with transcriptomics to get transcriptomic cell type together with activity. Also work in progress. But what I finally want to end with is merging brain with body and trying to understand brain dynamics and body dynamics in the same animal on the same footing. So the brain is very strongly connected to the body through, of course, hormones through the bloodstream, but also through the nervous system with big bundles of neurons connecting different organs to the brain. Looking at brain and body dynamics and behavior in the same data, in the same animal could be powerful for understanding the origin of some perhaps long time constants in the brain that may arise from the body, or spontaneous activity in the brain that may look random initially but may actually be due to changes in internal physiology, like changes in digestion or heart rate. So this is work by Virginia Ritten, who used to be a student in my lab and is now a postdoc in my lab, who's finishing up this project. Was busy finishing up this project, who created a fish with calcium sensor in all cells in the body instead of in all brain cells. And just by looking at the texture and shape of these cells, you can discern different tissues and organs. And here's a movie. This is sped up by a factor of 50, so it's not quite as fast as in the brain, but there are parts of the body that are really fast, like the peripheral nervous system, the muscles, and probably some other cells. But most cells have slower calcium dynamics. But there's still a lot of stuff happening. You can see here dynamics in the liver, gallbladder, kidney, gut, and so on. And all of these are bidirectionally coupled to brain dynamics, so that to understand brain dynamics, you really also want to take into account what happens in the body. Not all of it will be coupled, but we don't know yet without looking. So here's a top view, a top slice. You can see signatures here of neural dynamics and also astroglial dynamics. These are both captured. There's more motion, so you need to provide more alignment. But this is really more alignment algorithms. But the important thing is this kind of matrix. So instead of now having neurons over time, we have cells in all these different tissue types, including the brain, over time. And we could start looking for dynamic relationships between these different cell types. So I'll end there. I'm happy to chat more about any of this. But yeah, so the kind of way we want to go is to keep creating systems for ultra-observable experimental neuroscience and physiology, and through neurotechnology development, couple this to computational, statistical, and theoretical neuroscience. And by kind of pushing on both fronts at the same time, I think can allow for deeper progress. And we're very happy to collaborate with you guys on any of these questions. Just to thank En Young, who did the very talented postdoc, who's now faculty at UNC, who did the work on positional homeostasis. Yumu, also a very special scientist who is now faculty in Shanghai. Alex, who's a joint student between my lab and Florian Enger's lab, who did the work on connectomics and also on the molecular communication between astrocytes and neurons. Part of the electron microscopy connectomics team, Virginia, who worked on whole body imaging. Manuel, I didn't have time to present his work on spatial transcriptomics, and many others who I thank. All right, that's it. Thank you.
That never happens. Great talk. You mentioned briefly with the ketamines influence also on the zebrafish sort of resilience, but just without ketamine, do you feel like is there a mechanism that the animal will learn to be more or less resilient? Or essentially, with the model you show, like the threshold of N, how would that change?
It's a really cool question. I don't know. But it's a very nice question. Let me think if we have any data that speaks to that. So over time, we don't see much of a change. But the brain does what happens in the brain does change. So we see still a pattern of switching back and forth between giving up active and passive states. And the time constants don't seem to change. But the astrocytes in the brain, the first time they go from a normal environment to a helpless environment, we see widespread glial activity. If they are in a helpless environment for a while, we see more localized activity to the lateral medulla, where the circuit was concentrated. And that reduction in spatial footprint really helped us kind of identify these crucial cell types. So in the brain, something changes. In behavior, not so much. But it would be nice to see if there are ways in which animals could learn to become more or less resilient. We did try to teach them that being in a green environment meant that they were in a futile situation, and in like a red environment, it meant that they were in control. We tried things like that. In that case, it failed because they were extremely, like the color itself completely changed their behavior for some reason, which is, I think, an interesting question in itself. But that's why we couldn't continue that. But yeah, it would be really cool to look at that outside of.
Thank you.
Outside of ketamine.
Okay, thank you so much for the talk. I'm part of the team that is working currently on the Mu et al. Dataset, so I have a bunch of questions. But
on the glial dataset?
Yeah, on
cool.
neuronal glial dataset. But I guess one of the main ones is related to this question, and it's about this widespread activation, especially of the astrocytic network. Because if I understand correctly, in the proposed model, you have a quite localized mechanism where is this LMO astrocytes response as elicit this GABAergic neurons response. But then when you look at the data, there's this widespread activity of astrocytes. So first of all, what do you think is causing this widespread activity? So is it a signal that originates into the LMO and then is transmitted somehow within the astrocytic network? And then what's the potential purpose of such widespread activation? Yeah.
So the signal may come from the noradrenergic system or from astrocyte to astrocyte communication, and we don't quite know which one it is. We also see locus coeruleus activation, which lags NEMO a little bit, and so it's known that the locus coeruleus has projections very widespread across the brain. But there are also astrocyte potential pathways through perhaps release of ATP. I imagine that they also have ATP or adenosine receptors, or maybe ADP or AMP. So it could be through those extracellular molecular pathways or through gap junctions. Calcium could go through gap junctions. It could also be other messengers like cyclic AMP. So I don't know what contributes how much. Maybe propagation speed is something that could tell us something about that. So I lean towards thinking it's probably mostly norepinephrine. And then the purpose of the modulation, that's very interesting. We have another study that's ongoing where we look at the downstream consequences across the brain of this astrocyte activation. And there are changes in the way that circuits respond to visual stimuli, for example. So that's consistent with an overall kind of dampening of reactivity. But why there's this potentially dual pathway from sensory processing versus inhibiting motor output directly is an open question. And there also seems to be, so we know from prior work that this signal also spreads to the spinal cord. And now new work from Claire Vuillard's group has shown that this spinal calcium signal also directly inhibits spinal circuitry. So it seems to be kind of operating on multiple stages of the sensory motor transformation.
Thank you.
Yeah, so I have a question about the maximum number of attempts N. So can you estimate that number for each fish? And does it change over time? And how much does it change over fish? Are there any neural correlates to that?
Yes. So Over time, it doesn't seem to change much, but I should also say we haven't looked in great detail. Like, we haven't exposed the animal for a day and retested the next day. So it's a little bit anecdotal what I'm saying. When I've looked at these long data sets of several hours, I didn't see a clear prolongation of the passive period or so. I imagine that if you provide physiological stressors, there are stressors like salt, temperature, oxygen, carbon dioxide, and so that could shift the balance, and it would be very interesting. So ketamine obviously shifts it. We haven't seen obvious effects of learning, but yeah, we also haven't explored this enough. So I think mechanistically, what it depends on is how much norepinephrine is released, how many receptors the glial cells have, and the intracellular machinery for calcium release inside astrocytes. So any of these could change as a function of experience of physiological conditions, but we haven't systematically explored that space.